Hi there. So I'll be presenting this work that we've been doing developing energetic models that could be used to help upgrade the interim PCOD model framework, building on some work previously funded by Marine Scotland as well, where we focused in on harbour porpoises. This extends us to other species. And just want to acknowledge Magda, Katerina and John who worked significantly on these projects. The IPCOD framework is now 10 years old, so I think everyone's reasonably familiar with it. One of the things that many people understand is that parts of these relationships are very challenging and data limited. And so in the IPCOD framework, we've used expert elicitation to move from these changes in behavior and physiology to vital rates. But I thought what I would do is just summarize how the model has developed over the last 10 years and kind of where we are now. So with respect to disturbance, the expert elicitation functions were updated in 2018, and that significantly changed the model outcomes. Basically, the experts considered that the effects of disturbance were significantly lower than they were in the 2012 version of the elicitation. Experts also considered that considering 24 hours of foraging was overly conservative and so suggested 6 hours for porpoises and 2 hours for seals. Unfortunately, though, we didn't, in that elicitation, have time to cover bottlenose dolphins and minke whales, so the relationships are still those from the elicitation when the model was released in 2013. In terms of the effects of auditory injury, so permanent threshold shift, we upgraded the elicitation again in 2018, but again, we were time limited, so we were only able to focus on harbour porpoise, seals, and bottlenose dolphins, but did not cover minke whales. And so the minke whales we're still using the original functions. And the new elicited functions resulted in significant changes to the model outcomes as well. In terms of how the model is used in practical terms, currently being used on project level and cumulative impact assessments for offshore wind farm projects, primarily being put before regulators and decision makers in Scotland, Wales, and that's also being discussed in the Republic of Ireland currently. But I mentioned at the outset here we're focused in on some of the potential upgrades to these models in terms of how disturbance is modelled, and so I'll just talk you through some of the energetic elements. So hopefully everyone's reasonably familiar with energetics, but it's really just about energy in versus energy out. Animals forage on their prey, digest those prey and get energy from them, and then at different stages of life will dictate where they allocate that energy that has come in. In situations where energy is greater than the costs, animals can increase the sizes of their energy reserves, possibly in preparation for reproduction or to fuel towards growth. Of course, conversely, if they're expending more energy than they're consuming, then they will lose weight and eventually die. So that's obviously the kind of general principle of costs versus intakes. Single females are modelled over their entire lifespan, so they're producing calves and pups in the model simulations. And then in the case of porpoises and seals, the offspring are followed through to age 1 year. And then disturbance is represented as a ceasing of foraging for a certain number of hours, and I'll talk through that in a little bit more detail in a moment. So in 7 minutes, I would not do very well to describe 5 different energetic models, but suffice it to say, the models require a very large number of parameters, and those have all been sourced from the literature, but it's important to acknowledge that we're still data limited in many of these fields, and so there are uncertainties in these models and basically all other energetic models that exist, but it's worth being clear about those uncertainties. To get around that, what we've done is done sensitivity analyses to try and understand the impacts of the different values that were selected and to understand the key uncertainties. And then we've also developed an approach that allows the models to generate the kinds of transfer functions using energetic models that would replace those from expert elicitation. And this is all summarized in 2 reports that have been submitted to Marine Scotland along with the models themselves. So the other thing that we did in this project, and the other thing that's very important to using interim PCOD, is that movement matters. And in the context of disturbance, why is that? Well, so here we've got a population demarked by that outer green boundary. And then in this case, we've got a highly mobile species that moves around a lot within the population. You then impose a disturbance on top of that. And what you see is you basically see many of these individuals are disturbed, but relatively infrequently because they're moving all over their range. The other end of the spectrum is an animal where you've got more resident individuals or animals that are very constrained in their usage, And so what happens in this kind of circumstance is that you have a relatively small number of individuals getting a lot of disturbance. And these are two completely different outcomes when it comes to summing up into population effects. The effects on residents are significantly worse. And that's certainly what we've seen with these models writ large. So here, these plots basically show the effect of considering different disturbance effects on the y-axis. So animals getting 1 hour at the bottom of the x-axis, or 12 hours for every day of disturbance, and then different probabilities of being present to be disturbed, basically a proxy for different resident patterns. So highly resident species or individuals at the right side of the x-axis, and then highly mobile species on the left. And basically what we can see in conceptions, calf births, calf survival, and adult deaths, that basically it's really in these top right circumstances. So it's where you have high effects of disturbance, and you have a high probability of being repeatedly disturbed. Those are the key things that are driving disturbance. And so those are still things that we need to better understand as we use these models. But the models show that these effects of decreased foraging sort of follow what we expect in ecological theory, and it also tells us kind of what the key sensitivities are in using these kinds of models. So these are the kinds of things that we'll still need to be thinking about in PCOD research in general. So basically these DB models that we've generated for all 5 species can be used to replace the transfer functions derived for disturbance using expert elicitation. But we still certainly need to address deciding on what we think the effect of disturbance is in terms of how many hours of lost foraging occurs when animals are disturbed. And that may well vary over time and across individuals. So that's something that still needs careful thought. But these models can move away from the expert elicitation approach and replace those with these different kinds of functions. Again, it's important to be open about the fact that the model has other sensitive parameters that are within the model. So the thresholds for starvation and pregnancy, understanding when calves or pups become independent foragers versus how dependent they are on their mother, and then variations in daily energy intake, and also the effects of the wider environment in terms of prey quality and so on. And then in terms of writ large, what are some of the future improvements to IPCOD? One of these could be integrating these DEB models into IPCOD. As I mentioned at the outset, some of the older relationships from 2012, 2013 could be updated. And then there's hopefully stressed the point of movement, and understanding the probability of exposure and repeated exposure is critical. So that would be another area to try and improve. Thank you very much.
In this talk, I'll be presenting some recent work led by Matt Carter and myself, but involving many collaborators. So why do we care about seals? We have 2 seal species in the UK, the larger grey seal seen here at the back and the harbour seal in the front. And as you can see from this slide, the UK and Scotland in particular is an important population centre for both seal species. Seals are important at ecosystem level. Indeed, as integrators of bottom-up effects, they are used as indicators of marine ecosystem health, but they also have important top-down effects. There are multiple potential impacts of marine renewable developments, including on seal movements and hearing. The use of seals as indicators of ecosystem health, and more specifically, understanding the impacts of marine renewables on seals and the ecosystem, is dependent on knowing where seals go at sea. The large-scale at-sea surveys that are conducted for estimating cetacean distribution are not readily appropriate for seals. Thus, instead, we track individuals by gluing tracking devices to the fur on the back of their neck, and this falls off during the annual molt. As you can see, with our collaborators, we've built up a large database of GPS quality tracking data for both species around the UK and Ireland. Unfortunately for us, converting this to at-sea density estimates is not straightforward. Essentially, not all haulouts are visited by tracked animals. So we use the tracks to generate region-specific habitat association models. These allow us to predict density emanating from each haulout, whether it was visited by a tagged animal or not, with predictions weighted by the most recent haulout count. And combining across all our haulouts, we then generate a population level surface, which represents the percentage of the at-sea population for UK and Ireland per 5 kilometre cell. This slide shows the resulting mean predictions for grey seals, And we've also got the lower and upper confidence intervals, which I'll describe what these mean in more detail in a later slide. We've generated the same for harbour seals, and you can see that the harbour seal distribution is much more coastal. So for more information about the methods and to download these maps, please refer to the paper listed below, Carter et al. It's also important to note when you look at these maps that this is only the density that's emanating from seals hauling out in the UK. So it doesn't include the, UK and Ireland, so it doesn't include other parts of Europe like the Netherlands. Here I want to highlight that there are SAC specific maps available for download. These give predictions of density emanating out of each SAC. And for grey seals in particular, they're really important to consider. So for example, looking at this map for grey seals, you can see that there are high areas of density around the Isle of May SAC, which is in a white dot here. Indeed, 146 seals are predicted to be within the nearby NNG wind farm. However, looking at the SAC specific maps, we can see that very few of these 146 seals, 2%, are associated with the nearest SAC, the Isle of May, whereas almost 70% are associated with the SAC further south. It should be noted that the reason for this disconnect in the above example is that the Isle of May is an important breeding colony, but not a key haul out during the foraging season. And obviously it's the foraging season that the maps are based on. And essentially we can't link the breeding populations of an SAC with their at-sea distribution during the foraging season. Indeed, we estimated that between 20 and 60% of breeding females forage and breed in different regions. So in terms of the interpretation, we're often asked about the confidence intervals. What the confidence intervals are, here we've got the upper and lower, are the range within which we predict our average density to be, based on the habitat association models. And the mean prediction is simply the centre of that range. And as such, the confidence intervals, they don't encompass any temporal variation, say within or across days, which means the upper interval, the upper limit, doesn't represent a maximum density that could be in a cell at any one time. So these intervals are calculated on a cell-by-cell basis. So for each cell, what is the lowest or highest proportion of the at-sea population on average? As such, the totals for the lower and upper will respectively sum to well under and well over 100%. And for this reason, it's not straightforward to simply combine confidence intervals across cells. Having said that, for individual cases, we can generate confidence intervals at any scale of combined cells, and also make predictions from any combination of haulouts, like we have done for the SACs. I just want to briefly talk about the units. In terms of the use of relative abundance, which we often use, and absolute density estimates, which are often needed. So these are readily producible, which we have done in the most recent paper, using some scalars. But I would caveat that where possible, the relative abundance should be used. So if you're comparing between two areas, for instance. And that's because the second scalar assumes no temporal variation in the proportion of the population at sea at any one time. So for example, with tide or month. But also that there's uncertainty around those, the means of the scalars aren't propagated into the uncertainty around the density maps at present. This also leads me to highlight, in terms of density, that it's not an indicator of the number of individuals or the proportion of the population that use a cell overall. So in this example here, where we've got the haul out shown as a yellow star, we've only got 20% of the abundance within the cell that encompasses that haul out. However, every single one of these animals that are away at this foraging in this the cell to the right must go through these cells to get to the haul out. So I just wanted to highlight that. In the interest of time, I'm not going to go into this in detail, but it's just to highlight that there are in the short term updates coming that will incorporate the recent Shetland tracking data, and also to update the abundance estimates from the haulouts. There are also longer term requirements to increase accuracy and improve the uncertainty estimates, and also to extend this to a wider spatial extent. It just remains for me to thank ScotMER, my colleagues at St Andrews, collaborators and facilitators elsewhere, and also lots of different funders, which include those shown here. Thanks.
I'm Gordon Hastie, and I'm a researcher at the Sea Mammal Research Unit at the University of St Andrews. And I'd like to talk briefly about some of our work into seal behaviour around tidal turbines. The work's very much a team effort and represents a lot of hard work by a lot of people, particularly my co-authors here. The project was funded by NERC and the Scottish Government and represents a very close collaboration with the tidal developer CMEC Atlantis Energy. Spatial and temporal overlap between tidal development areas and marine mammal distributions has led to concerns about interactions between animals and turbines, and in particular the potential for fatal collisions with rotating blades of turbines. However, until recently, data to quantify these collision risks has been almost completely lacking, and in particular data on the behavioural responses by seals to operational turbines has been a significant data gap, and as such has been one of the key knowledge gaps in the ScotMER marine mammal evidence map. We've now developed a reasonable evidence base from tagging studies showing that seals exhibit some degree of avoidance to operational turbines at scales of hundreds to thousands of metres. For example, this figure in Joe Nufrio's recent paper shows the estimated scale and extent of avoidance by harbour seals to the operational May-Gen array. However, data on the fine-scale movements of seals in close vicinities to operational turbines at scales of metres and seconds remains a key data gap. And part of the reason behind this lack of data has been the challenge of tracking seals underwater at the spatial and temporal resolution required. So to address this, we developed a marine mammal high current underwater platform through a NERC innovation grant a few years ago. Broadly, this system is a seabed platform that houses 2 multi-beam imaging sonars, a small array of hydrophones, and a series of control electronics. The system was designed to receive power from an umbilical cable connected to the turbine and uses fibres in the turbine export cable to relay data in real time to computers on shore. And the rationale behind this was that it would give us the capacity to collect data over the periods of many months or even years and collect sufficient numbers of encounters between seals and turbines. And I'm delighted to say that after a number of false starts, in May last year, we successfully deployed the platform next to one of the operational turbines in the May-Gen array. This was carried out by May-Gen engineers as part of their routine servicing and deployment activities. I hope we can just about see our platform being lowered into the sea there by the crane on this rather oversized North Sea giant. And this was lowered to a location 30 metres to the north of the turbine and was oriented so the sonar is pointed directly towards the turbine. And what we're looking at here are data from the 2 sonars. We're effectively looking at a plan view of the turbine. You can see the turbine in the middle of the frames here, approximately 30 metres away. And the vertical orientation of the 2 sonars is offset by approximately 15 degrees. And this means that we can cover the entire water column with the sonars, but it also gives us means of estimating the depth of the animals as they move past the turbine. The sonar on the left, you can see is the upper sonar, and you can see the strong target in the middle is the nacelle of the turbine, and you can just about see the rotating blades of the turbine there. The one on the right is the lower sonar, and you can see, you can't see the nacelle quite as strongly there, but you can see the foundation as a very strong target, and you can see elements of the seabed around about the foundation there. We've been running the system now continuously for a period of almost 9 months. And the good news is that we've detected a very large number of animals moving within the sonar data. And most importantly for us, we've now detected a reasonable number of seals moving past the turbine. This is an example here of one of our seals in the sonar data. You can see it comes in approximately 15 to 20 metres on the left and moves across to the right there, between the turbine and the sonars. Something to note here is that this seal was only detected on the lower sonar, indicating that it was relatively close to the seabed as it passed. And very interestingly from an ecological perspective, but rather frustratingly from a seal tracking perspective, we're also frequently detecting quite a large number of other mobile species close to the turbine. These include a range of different elasmobranch species, large schools of fish, and also diving birds. But in terms of turning these kind of raw image data into what we need to analyse seal movements in relation to the turbine, Doug Gillespie here at SMRU has done a fantastic job developing the software PanGuard to visualise the data and run a series of movement detectors in the data. This writes the results of the detectors to a database and a series of binary files, and it also has a really nice sonar interface that allows us to quickly review and allocate an ID to targets of interest and provide a nominal confidence score to each of the IDs. At the moment, this is a relatively user-intensive process, but at this early stage, we feel it's a conservative approach, and we want to ensure that we're not missing anything critical before working on implementing automated classification approaches. So to summarise some of the early results from the data we're getting in. As I mentioned, we're getting good numbers of targets detected by the sonar, and most importantly for us, we're getting almost a seal a day in the data. We're also getting good numbers of these other species, including diving birds, fish schools, and we're also getting relatively high numbers of relatively small targets which remain unidentified. And you can see the associated confidence levels that the auditors have allocated the different target types here, from birds on the left to fish in the middle and marine mammals on the right here. Ultimately, though, the key data are going to be the tracks of individual seals past the turbine. I've plotted all the seal targets for a 2-week period here at the start of the data collection. And a couple of things to note. Firstly, you'll see that some of the tracks appear exclusively in the upper sonar, some in the lower sonar, and some in both of the sonars. And these are the kinds of data that allow us to estimate where in the water column the seal was as it moved around the turbine. Secondly, you'll see that there are either gaps in the tracks or points where the tracks effectively disappear. And this is likely to be an artifact of the targets dropping briefly below the acoustic intensity threshold in the PanGuard detector. So to recreate the full tracks, we clearly have some additional work to do post-hoc. So in terms of where we'd like to go with this, we'll be continuing the data collection until May this year, completing a full 12 months of data collection. We hope to use the annotated data from the audits to develop a series of new machine learning algorithms for the data. We then need to fill in the 3D seal tracks and match them to the operational status of the turbine. But ultimately, the key thing I think we need to think about is how we turn these kinds of 3D tracks of animals around the turbines into metrics suitable for the kinds of collision risk modelling approaches that are being used by industry and regulators. And with that, I'd just like to say many thanks for listening.
Hello, my name is Susanna Quer, and today I'm going to give a run through the acoustic monitoring projects that Marine Scotland Science has been involved in over the last 10 years, and update you on our desired plans for the future. All the projects that I will mention have in common their methodology, the use of passive acoustic monitoring, through moorings characterized by 3 pieces of equipment: a cleat detector, a sound recorder, and an acoustic release. Due to battery and storage constraints, these moorings are serviced 3 times a year with only a subset of moorings left in place over the winter months. The first of the projects that I will be talking about is the East Coast Marine Mammal Acoustic Study, or ECOMMAS. It has the longest running cetacean acoustic dataset at Marine Scotland Science. It started in 2013, so that's over 10 years ago now, by our team leader Kate Brooks, to examine the broad-scale effects of offshore wind farm construction in the East Coast, focusing on the protected population of bottlenose dolphins, but also on harp porpoise. It consists of 30 monitoring stations distributed along 10 locations at the east coast of Scotland, at 5, 10 and 50 kilometer distance from shore. With over 600 deployments to date, this is, as you can imagine, a huge amount of acoustic data collected. Currently, we are amalgamating these 10 years of data to look for any trends in porpoise distribution. However, data from ECOMMAS has already been used in different projects throughout all these years that had led to the publications that I'm showing on this slide. It has also helped on masters, and it has been used for PhD projects. Not only focused on these 2 species that I mentioned, but also on minke whales or on projects focused on underwater noise acoustics. In fact, the ECOMMAS array has also made possible for us to contribute to the EU-funded project JOMOPANS. This project combined modeling with measurements to provide a management tool for underwater noise monitoring in the North Sea. 2 of the ECOMMAS locations provided data in the area and have now become key for us to report underwater noise measurements under the MSFD requirements. Marine Scotland Science is also contracted by Seagreen and NNG offshore wind farms to deploy moorings in the wind farm sites, the location of which you can see on this map. This data is then analyzed by SMRU Consulting to help monitor any changes that may have occurred in the station distribution and background noise levels in the area over the course of the construction phase. Now, moving over to the West Coast, we have contributed to 3 different projects. The first one is COMPAS. This was an EU-funded project run in close collaboration with SAMS and AFBI, the Agri-Food and Biological Institute in Northern Ireland, from 2017 to last year, 2022. The focus was to develop a long-term monitoring strategy to address marine mammal distributions and the level of underwater noise in this cross-border region between Scotland and Ireland. From a total array of 10 acoustic moorings routinely deployed, 6 were serviced by us at Marine Scotland Science, seen here in purple on the map. The data from COMPAS project has already materialized into different reports, into a publication, and it has been used into different PhDs and master theses on marine mammal presence, but also on underwater noise or even on equipment performance. The other West Coast project that Marine Scotland Science has been involved with in the past years has been MARPAM. MARPAM is also an Interreg EU-funded project, but in this case it was set up to provide evidence for the 2 species of seals present in this cross-border area for the regional MPA management plans. MARPAM actually helped fund the updated at-sea distribution of seal maps. While at the same time, 7 more moorings were deployed. They were all complementary to COMPAS, and 3 of those moorings provide the first in situ measurements of underwater noise in the West Coast, which actually were compared to the first modeling of underwater noise in the West Coast that was produced by Jonah's Interreg project. Finally, the SAMOSAS project, an EMFF-funded project coordinated by MSS and commissioned to SAMS. The array of 10 moorings deployed under SAMOSAS help collect year-round data on seasonal distribution and diurnal activity patterns of cetaceans in the region west of the Outer Hebrides between 2020 and 2021. The results can be found in the published article Monitoring Cetaceans Occurrence and Variability in Ambient Sound in Scottish Offshore Waters by Ninki et al. All these projects that I have been talking about have produced a series of outputs that increased our understanding of cetacean distribution patterns, as well as the levels of underwater noise in both the west and the east coast of Scotland. This data has been used to assess how cetaceans could be affected by anthropogenic activity, as well as informing marine protected area management plans, helping us to initially address several ScotMER knowledge gaps. It has also created a legacy of data that can be further interrogated to expand our knowledge and help advance the passive acoustic monitoring field for its use on marine mammal science. As of today, all these large-scale acoustic monitoring projects on the West Coast are now completed, while ECOMMAS is hopefully set to continue. This occurs at a time in which the Scottish Government's ambitions to achieve net zero by 2045 has led to the first Scotwind offshore leasing round, which is set to become the world's largest commercial round for floating offshore wind. These leasing areas are further offshore and in areas we have not acoustically monitored before, or in which we know little about in terms of cetacean distribution and abundance. Given this scenario, the strategic monitoring objectives in line with ScotMER as well as our policies, at Marine Scotland Science we would like to expand the ECOMMAS array. We want to continue monitoring in some of the locations on the west, as well as expanding further north and into the east. We call this expansion the Scottish Passive Acoustic Monitoring Network, or SPAN. We understand that the caveat of acoustic monitoring would still be present, that is that the spatial coverage of our monitoring would be low compared to other methodologies. However, acoustic monitoring provides not only all year-round data, but also data during the night or under bad weather conditions, time at which other methodologies are very unlikely or not possible to occur. This kind of dataset is therefore key to assess more truthfully the presence of marine mammals or the impacts on any further marine development in Scottish waters. Thank you very much for listening, and thank you very much to a large team of scientists, technicians, and boat crew that have made all of this possible. Thank you.